Okay, so hi, I'm Oscar. I'm a postdoc in UMass Chan Medical School and Harvard University, and I'll be hosting today's Science Cafe session. And today we will be discussing an interesting topic, injury and regeneration. And we will have two speakers. Our first speaker is Carlos Rivera-Lopez. And Carlos is a PhD candidate in the Srivastava lab in Harvard University. And he studied adult pluripotent stem cell of Hofstad and Miami, and you're gonna learn that in his presentation. And in his spare time, he likes to introduce the Latin Society and into the STEM field. And Carlos.
Alrighty, thank you so much, Oscar. Let me share my screen. Okay. Well, thank you everyone for joining. It's a beautiful evening, so I'm thankful for everyone who decided to connect and learn a little bit more about the stuff that I do in the lab, which has to do with regeneration, and like the title of my presentation, how to grow a new head, which is a crazy question to ask and even consider, given that a lot of animals like us cannot do this, but there are certainly some animals that can do it, and we're going to learn about one of them in this presentation. Like Oscar said, my name is Carlos. I'm a graduate student here in the Srivastava lab in Harvard University, where we study regeneration and in general how injuries heal over time and what we can learn about that when we think about human health and human treatments when injuries happen. But before we get into it, I wanted to open a poll, which one of my colleagues here in the presenters are going to share. Here it is, just to get in the sense of how much we know about regeneration or what we consider regeneration. I figured I could ask just a question: which of the following options is considered regeneration? And if people want to chime in, either by saying something or just voting in the poll. Love to see that everyone is considering everything above: replacement of cells in the inside of our intestines, when a shark loses a tooth and then it regrows it back, or the hair that grows from hair follicles in mice, or the fourth option, which is all of the above. So everyone answered. Most of you chose all of the above, which I just read, which is the correct answer. All of these are considered regeneration, just different capacities that some animals have. Stop sharing this. So being this said, we can think about regeneration as this capacity to regrow and replace tissues, be it tissues, cells, organs, over time when an injury happens. And if we consider regeneration as a spectrum, we can see that we can organize animals in the spectrum depending how much or how little they can regenerate. If we consider animals in the least end of the spectrum, which are the animals that can regenerate the least of them, we see that we fall in here. We can regenerate very specific parts and tissues of our bodies. And I just wanted to give a quick example of one of them, which is one of my favorite examples in just human regeneration capacity, which is the regeneration that occurs inside of our bodies, specifically in the intestinal lining. This intestinal lining is referred as the lining that falls inside of our intestines, which needs to be healthy and well-functioning for our body to absorb nutrients and also remove the waste. And the way this lining is kept healthy and well-functioning is by the development or the regeneration of new cells covering that lining over time. And this works in the way that cells that lie in this little crypt will proliferate, divide, and then move to the outside of the crypt, which is the actual lining of the intestine, where these cells are mature and undergo all the functions that the intestine do. But this mode of regeneration in which cells divide, mature, and then move out, it's a cycle that happens periodically inside our bodies. And this is just one of the many examples of regeneration that occurs in our bodies. Muscle gets regenerated, our hair follicles get regenerated, cells in our skin also get regenerated over time. But there is a big limit to the type of regeneration we can do, which is, for example, we cannot grow a whole new arm if we lose one. However, this is not a limitation for other animals that lie in the center of the spectrum of regeneration, like, for example, lizards and salamanders. They can very well regrow limbs or tails when they lose one. And they do this without any issue, but what they definitely cannot do is grow a new head. This is the limit, and this is a limit both for salamanders and also for humans, as we know, because regrowing a new head is reserved for animals that lie on the other end of the spectrum, where I am defining this type of regeneration as whole body regeneration. Animals on this end, they're able to regenerate all of the parts of their bodies when they lose one. And I'm highlighting this animal here as one of these animals that can do this. This animal is the main protagonist of this presentation, also the main system that we study in the lab. It's called Hofstenia miamia, and it's commonly known as a three-banded panther worm. Three-banded because of the three cream-colored bands that you see across the body, and panther because, as my advisor likes to say in her presentations, this is a voracious predator that likes to prey on smaller animals in the wild. And I say smaller animals with the big caveat that these animals are pretty small themselves. Here is a video. I am moving them using a plastic pipette in a petri dish. You can see that they swim in the water, but they're pretty small. And in order for us to appreciate all the details and study them in the lab, we need to work with them under the microscope. These animals were introduced into science and laboratories over a decade ago. These were collected from the Bermuda Islands, which is just a normal scientific process that happens when you want to establish new animals and new systems in a lab. You collect them, you bring them over, and lucky for us, we can maintain this a culture of these animals in the lab, which we can reproduce and keep alive to make good science the way we do. But I made a statement at the beginning of the presentation, which is that this animal cannot go whole body regeneration. And just so we can grasp what this means, here I am showing you an uncut version of this animal. And if we induce this artificial amputation where we cut this animal in half, we see that the animal, after 1 day, is able to close up that wound, both in the original head, this wound is already closed, in the original tail, that wound site is also closed, which is a big stark difference when we compare this type of healing with other animals, because if we suffer a big injury where we cut ourselves in half, none of our two remaining fragments are going to survive. And this is very well true for a lot of animals, including axolotls or other salamanders that can undergo bigger regeneration or more robust regeneration than we do. But what we find is that over time, these fragments are now regrowing what they lost. You can see that this original head is now growing this pointy tail here, which is white, is unpigmented. This is new tissue that is growing from the wound site, as well new tissue growing from the tail fragment, which in this case is going to eventually form the head, including the brain, including the mouth, including all the cell types in here. And there is something special about this tissue that is growing from both wound sites, and I want you to pay attention to that. Because if we ask what is special about this new tissue that is growing from here, if we schematize this to just simplify things here, we see that over time there is an outgrowth in these wound sites, and we call this a blastema. This is again an unpigmented tissue that will form the basis of the new tissue when regeneration happens. The blastema is important. So in the absence of the blastema, if we remove this blastema from the wound site, there is no regeneration going to be formed. But once regeneration happens, we see that both fragments are going to regrow everything that they lost. Muscle, which is highlighted here in green, will be reconstructed in both fragments. The brain, which you can see here in magenta, will be grown if needed. For example, a tail fragment growing a new head will grow a brand new brain, whereas an original head that has the original brain will no longer make a new brain. Instead, will make the remaining cell types, which include the digestive system. This is something that will be in both fragments, as well as the epidermis, which is here highlighted in blue, surrounding the whole animal. This is regrowth in both fragments. But what about the blastema is so special about this? Why this outgrowth over time actually becomes a whole just functional animal? And if we focus in this cell fragment, we zoom into the blastema, we find that there is a special type of cell found here in the blastema when regeneration is happening. These animals, these cells that are in the blastema are historically known as neoblasts. And these are stem cells or a special type of cell in this animal that is the one in charge of reproducing, proliferating, and then forming the tissues that were lost when regeneration happens. These neoblasts, again, are found in the blastema, and if we remove these neoblasts, we kill these neoblasts when regeneration is happening, we see that regeneration is hindered. There's no regeneration happening. So there is something important in the blastema, specifically the neoblasts, that allow regeneration to happen. So the lab, we've been focused in understanding more this population of cells, and we found that these neoblasts in the blastema are pretty different to themselves. I'm highlighting here false coloring these neoblasts, so you see the diversity in them, but you can see a pattern in the sense that there's only 4 colors and there's more than one cell with the same color. And that means that although they're different among themselves, there's something in common between them. And something that makes them similar, for example, is the types of genes they are expressing. If we focus specifically, for example, in the ones in pink, I'm highlighting some of these neoblasts in pink because these are neoblasts that are expressing, for example, say, genes that are important for the function and the just well-functioning of neurons, for example, in the brain. But because these genes are now being expressed, are actively being expressed in neoblasts, which are not neurons, we hypothesize that these are the cells that will eventually form the neurons. So again, because these genes are expressed in cells that are yet to become neurons, we hypothesize that these are the specific type of neoblasts that will eventually form the neurons. And this is also, we hypothesize that this is also true for the other cell types, where, for example, neoblasts in yellow are the ones expressing genes that are found in the digestive system. So we hypothesize that these are the neoblasts that will eventually form the digestive system. And this, I agree with you and everyone I have a conversation with. This, although it's a hypothesis and it looks pretty straightforward, it looks logical that something that is expressing a gene that seems to be important for a cell type, and eventually when regeneration happens, that functional tissue gets remade, then it's a good hypothesis established the way that we see the neoblasts forming the specific functional tissues. But although this seems pretty straightforward, we need very robust, very specific data showing that a neoblast that is expressing muscle genes is eventually forming the muscle. So I am at that stage of my PhD where I ask myself, what kind of tests or techniques can I use to really show that a neoblast, when regeneration is happening, is forming specific functional tissues? And this is where I introduce this very cool and exciting technique that we have been able to establish in the lab. It's called transgenesis, and we use the same information from those genes that are found in the neoblasts to drive the expression of fluorescent proteins. I'm going to rewind this video a little, and you can see here 2 different animals. One of them, here, this one in the bottom, you can see that it is bright in specifically where the cream color bands are. You can see them here, you can see them here, here. And this is called autofluorescence. This is a type of autofluorescence, a fluorescence that exists naturally in the animal. But what I want you to focus on is in the other animal, this conglomeration of cells here, which is forming the muscle cells in the animal. These muscle cells naturally will not be bright, will not be glowing green, And they are growing here in this animal because we've used this technique to label the cells using proteins that are fluorescent under green light, and we're able to track those cells in the animal. And the idea with my project is that we're going to label specific neoblasts and then see them as they regenerate, as the animal regenerate, where are those glowing neoblasts appearing, and whether if we're expecting a green neoblast, which is representing the muscle neoblast, eventually forming a muscle, if it's true, then we can tell that our hypothesis our expectation was met based on our hypothesis. The way we establish a transgenesis is by injecting a specific injection mix that contains all the reagents needed to label these cells in Holstenia, in the worm. And you can see here in this video that I am using a setup that is to my right. You can see it here. This setup is holding the needle that I am going to use to then inject the embryos with the injection mix to label those cells. So here I'm representing, this represents the embryo, this is the needle, and this is just the first step in the process where I'm injecting the embryos to later on be able to see specifically glowing cells from those that I'm interested in tracing. And again, I want to go back to this slide and highlight that the important aspect of this is that I'm trying to understand the dynamics that happens in these arrows. I'm trying to ask, what about these cells that lie here in the blastema? What are they doing when regeneration is happening? What sorts of stages are these cells going through to then form the specific functional tissues that were lost? So this is my overarching question within the lab. These are the techniques that I'm using to ask these questions. So now I am happy to take all questions as needed. Here is another video of the animals in the lab. So I'm happy to take any questions about the research, about being a grad student, understanding a little bit more about the work that I do and other folks in the lab also do.
Thank you so much, Carlos. That was a fantastic talk. So we'll now proceed with the questions. So if anyone has any questions, please type them in the chat box and we will ask them for you. So the first question is, does the tail piece grow back slower than the head piece?
That's actually a good question. I just want to clarify something. When you say the tail piece, do you mean the original tail forming a head?
Yeah, I believe so. I believe that's
okay,
what, yeah, unless the person wants to clarify, but that's how I'm reading the question.
Right, cool. So that's a very good question, a very cool question. We see when we, for example, cut the animal in half, you have the original head, which presumably will have to regrow a less complicated tissue because it's only the tail. It doesn't hold, for example, the mouth or the brain. So we see that our growth of the tail happening quicker than the head will grow back. And we sort of have this hypothesis that actually that regeneration doesn't maybe require as much proliferation as the tail fragment forming the head, because essentially that's just closing the wound site and then just forming the pointy tail. And, you know, maybe muscle, maybe epidermis, pre-existing muscle and epidermis are enough to just close up and then form the tail. Eventually, when the worm resizes and, you know, grows to the proportion of the original head, yes, proliferation has to be involved, but that quick close-up of the wound and then growth of that pointy tail happens quicker than the regeneration of the whole new head. That's a great question.
Very cool. Thank you. The next question is, are there any specific genes that are differentially expressed that help them have high number of stem cells?
That's another good question. This is ongoing work, not only from our lab, but also other labs. The stem cells in our system are marked by the expression of specific genes that have functions in the germlines of a lot of other animals. All of us have expression of those genes in specific cell types or specific stages of our bodies, but these animals are able to maintain that expression of the genes within the stem cells at all times in their lifespan. These are adult animals that are reproducing, that are remaking all their tissues, even if injuries are not happening. And we believe that the expression of those genes in those cells are important to maintain the identity of the stem cells and the functionality of them.
Great, thank you. Our next question is, this seems like a great evolutionary benefit for these organisms. Do they reproduce in high numbers as well?
These animals do reproduce a lot in the lab. It's one of the advantages we have in the lab is that they reproduce and they lay eggs, which are the embryos is what I use to make the transgenic animals. So in the absence of the embryos, transgenesis would be a little bit harder to establish in the lab. So yes, they do reproduce a lot. There are other animals that can do whole body regeneration, but instead they are asexual. So their main mode of reproduction is by fissioning themselves, by breaking them apart and then expand clonally.
Very cool. Thank you. The next question is, are the neoblasts that express cell type specific genes in the blastema already pre-existing in the intact animal before surgery, or are they generated after surgery from less specialized pre-existing neoblasts?
Those are two great questions. The first one, the answer is yes. Those cell type specific or the neoblasts that seem to be primed or specialized for a specific lineage, those do exist in the intact animal in way less proportion or density than the ones regenerating. Once you do the amputation, you're inducing the worm to make more of those neoblasts to proliferate and form the new tissues. So we're artificially increasing the numbers, but yes, we are able to capture those same cell types during homeostasis in the absence of an injury.
So I just want to clarify with the follow-up. So are you saying that there is already 4 types of cells that are sort of found throughout the body of the animal ahead of time, or is there one kind of stem cell that sort of differentiates after the injury? Oops, sorry.
We see the stem cell compartment, the neoblast compartment, as one containing all of these cell types. We have the more specialized neoblasts that we believe are sort of progenitor-like cells that will eventually form the differentiated cells. But we have another subset of these neoblasts, which is another big hypothesis that we have in the lab, which is labeled and it's expressing specific markers that are not found in other neoblasts, which is forming the less specialized, the more pluripotent neoblast. And we believe that that is the one in charge of then forming the progenitors, which is the more specialized neoblasts. And I think that also tackles the second question, which is, are they generated from less specialized pre-existing neoblasts? That's the main hypothesis we have in the lab, is that there is a subset in charge of forming the progenitors. But again, to really establish those conclusions and see those results, we really do need the transgenesis and be able to track a given less specialized neoblasts, then forming specialized neoblasts, then forming differentiated cell types. So these are great questions, and they are very specific to my project. So I'm glad to read them.
One final question. So has anyone looked at genetic differences between animals who can regenerate like this and those who can't? And has anyone figured out the genes associated with this? Are they evolutionarily conserved? Probably not, because obviously either they evolved at some point or they were there and then they were lost, or is that known?
Yeah, that's another good question. I think you can see conservation at different levels. If we focus, for example, in animals that can undergo whole body regeneration, what we find is that stem cells in each of these systems are heterogeneous in the sense that the stem cell compartment is composed of different subsets of those stem cells. Another conserved thing that over time people have been finding is that the more specialized stem cells will have the expression of differentiated cell type markers, as well as the stem cell markers. But all of these these characteristics or these features combined are not found in animals that cannot do whole body regeneration. So for example, if we talk about humans, we do not have a stem cell compartment that is able to form all of the cell types in our bodies. We do have specialized stem cells that are the ones in charge of, for example, forming the skin, but a skin stem cell cannot form muscle cells or cannot form neuron cells, which is a stark difference from animals that can do whole body regeneration. So yeah.
Great, excellent. Thank you so much.
Of course.
We can now proceed with the second talk.
Thank you for the questions.
Thank you.
Okay, thanks so much, Carlos. So we'll get to our next speaker, Wenjia Huang. Wenjia Huang is a 50-year PhD student at Alex Burns' lab at UMass Chan Medical School, and she studies axon regeneration in C. elegans. Wenjia grew up in China and attended college at UC San Diego, and she was majoring on human biology. And in her spare time, she likes painting and trying different kinds of food. And she will talk about axon regeneration in C. elegans. So Wenjia.
Thank you. Can you guys see my screen okay? Right.
Yes.
Yes.
Cool. Thank you. Thank you for inviting me. I'm very excited to share my research with you tonight. Charles has done a wonderful job introducing regeneration of different tissue types, and my work specifically focused on repairing the cells in the nervous system called neurons using a different type of worm, C. elegans. Neurons are information messengers. They receive information, integrate it, and pass it along through this long wiring process called the axons. And you can think of this information flow as a relay race. When the flow is disrupted by injuries, the information can no longer get where it needs to go. And you can imagine how severely the injuries can impact our abilities to perform daily tasks. Luckily, certain type of neurons can repair these damages themselves through a process called axon regeneration. And during this process, the part of injured axons that can disconnect it from the cells will fragment and eventually being cleared away, versus the part of injured axon that still connect to the cells can initiate a hands-like structure called the growth cone, which will regrow and hopefully reform a functional connection with the appropriate target cells. However, not all neurons can regenerate. For example, the neurons in our brain and the spinal cord have a very limited capacities to cell repair. And currently, there's not effective drug treatment to treat injuries in the central nervous system, leaving the damages to these nerves currently irreversible. And my research aimed to understanding how axon regeneration occurs in the neuron type that can cell repair and apply that knowledge to hopefully promote axon regeneration in the neuron type that normally cannot cell repair. And in the burn lab, we use roundworm C. elegans to study axon regeneration. The worm genome is highly conserved. At least 60 to 80% of human genomes genes are conserved in C. elegans, and therefore our findings in the worms are also likely relevant in humans. In the lab, we can use a laser to injure the neuron type of interest and study how animals respond to neuronal injuries. And as you can see here, because the worm has a transparent cuticles and a very small nervous system, only 302 neurons in worms, we can study axon regeneration in the real time with single neuron resolution. And in these time-lapse videos, you will see we injured the first two axons. The injured axons on the left is able to initiate a growth cone and eventually grow back to its original places, versus the injured axon in the middle failed to regenerate. And here I would like you to make a guess. How fast do you think the axon regenerate in C. elegans? And you can type your questions in the chat box. Do you think they can regenerate half of days, within after a day, or a week? Okay, I see responses coming up. Right. Any more guesses? So far I see people say all of the options. Some people say half day, some people think it's 1 day, and I also got one response that's 1 week. All right, so the answer is that the worms is B. The worms can initiate axon regeneration a day after injuries, which is really cool. The axon not only regenerate and regenerate really fast, and in wild-type worms, about 60 to 70% of injured axons can initiate axon regeneration a day after injuries. And with these awesome models, our lab recently found through candidate approaches that injured axons can regenerate better when we inhibit a group of enzymes called poly-ADP ribopolymerases, and it's a lot easier to just call them PARPs. So PARPs affect protein functions by adding ADP ribotrans to the substrate proteins, and this process is called PARylation. In the absence of these PARPs, we can see that more injured axons can regenerate in C. elegans. And this poly-ADP ribose of PARylation is reversible and can be later removed by a different groups of enzyme called PARCs, or poly-ADP riboglycohydrolysis. Without the PARCs, we see the opposite effect in C. elegans, that less injured axons can regenerate in the absence of PARPs. And together, these two results suggest that the protein PARylation is very important to regulate axon regeneration in C. elegans. Very excitingly, the function of PARP to regulate axon regeneration is conserved. We also see more axon regeneration occurs in the mice neuron cultures when we knock down PARP1. And next, we want to investigate in worms whether the regenerating axons are functional. So without injuries, the wild-type animals can move forward in these wave-like motions, and we can record their motion traces in a lab with a camera. And this wave pattern is caused by the alternative contraction and relaxation of the muscles along the worm belly and back. When we injure axons that are required to relax the back muscles, the worm will start to move in circles. And these circular motions continues in wild-type animals even after axon regeneration occurs, suggesting that the regenerating axons in the wild-type animals are not completely functional. Interestingly, in the animals that lack of PARPs, we start to see the worms stop moving in circles and regain their pre-injury motions after axon regeneration occurs. This suggests to us that perhaps the PARP loss of function animals are able to form more functional connections called synapses with muscle cells compared to the wild-type animals. To visualize these functional neuronal connections, we can do it in the lab to label the neuron in red and the synapses in green. And then we can measure whether these neuronal connections are properly reformed in part by counting the number of green dots before and after injuries. So in the wild-type animals, after injuries, the number of green dots reduce and is not able to restore fully back to the pre-injury levels, even after axon regenerate all the way to their original target. And data is quantified here. And this result is consistent with our previous worm tracing results that the regenerating axons in the wild-type animals are now fully functional. In the absence of PARPs, after injury, we initially also observed the similar reductions in the number of green dots, but very surprisingly, after axon regeneration occurs in the PARP loss of function mutants, we start to see the increased number of green dots that is comparable to the pre-injury levels. This means that the PARPs are actively preventing regenerating axons from forming synapses. And this is probably explain why in our previous results that in PARP loss of function animals, the animals are able to regain their function. So together, our result shows that the injured axons can regenerate better when we inhibit PARPs by both promoting axon regeneration and neuronal connections in regenerating axons. It also shows that forming synapses with target cells is not a secondary consequence of axon reaching to those targets. Instead, both axon regeneration and synapse reformations are carefully regulated after injuries. And currently, we are very excited to further expand our knowledges on how PARP inhibit functional axon regeneration. If you are interested in my work and our researchers, please check out our lab website, send us an email, and contact Dr. Byrne for more research opportunities. And with that, I want to thank you for your attention, and I can take any questions from now.
Wow, that was a fantastic talk, Wenjia. Thank you so much.
Thank you.
The first question we have is, what's your neuron of interest and why? And also, is there a behavioral assay to confirm injury?
And that's a great question. So in our lab, for my project, I studied the GABAergic motor neuron, which is controlling for the muscle relaxation. And there's indeed a behavior assay we can confirm the injuries. So if you injure the majority of the inhibitory motor neurons and attach the worms, they are not able to reverse in response to targets. And that's the way we know that our injuries is very efficient to completely disrupt the functions before axon regeneration occurred. And then we can also use the same injuries, same assay to measure the degree of connections.
Very interesting. Thank you.
Yeah.
The next question is, if you cut multiple axons, can all of the axons regenerate?
That's also a good question. So if we, for example, if we cut the majority of inhibitory motor neurons in elegans, we again see about 60 to 70% of injured axons within the same worms that initiate axon regeneration.
Interesting.
And in the PAR mutants, the number of regenerating axons is higher, which we are really excited about.
Thank you. So the next question is, how about sensory neurons?
So in elegans, there's also a group of researchers done on the mechaninsms neuron called PLM, and they show that in that neurons, after injuries, they actually have two ways to repair themselves. One way is through axon regeneration, as introduced in this talk. Another way is that those injured axons can actually fuse after injuries and also have functional restoration, which is very cool.
Oh, so they fuse after injury. That's interesting.
Yeah, just like fuse back together. Yeah.
Okay. The next question is, can you speculate as to why axon regeneration is not optimized in the wild type? In other words, why do we need to knock out PARPs to allow the animal to regenerate their axons better?
Cool. So my answer to this question is that I think axon regeneration is not optimal process because, as you can imagine, after development complete, where all the guidance cues are no longer present, this axon regeneration process is error prone. So potentially, the regenerated axons can land on the other targets it's not originally connect with. So that's why in wild type animals, axon regeneration is not optimal. And then perhaps there's a mechanism actively inhibiting this process. And then by identifying those targets, perhaps we can promote axon regeneration. Of course, we need additional methods to make sure those regenerating axons are properly guided to the correct practice in clinics.
Interesting. So you mean to say there might be misfiring or something just because of that? Okay.
Yeah.
The next question is, do the males, sorry, I didn't get this one. Do the males do the axon regeneration similarly?
That's actually a great question. So the worms that we study are hermaphrodites, which means they can self-propagate. They have both the sperm and oocyte together, at least in the work I'm doing. I'm pretty sure some people done axon regeneration in the male worms specifically, but I don't know the knowledge on top of my head right now. I will get back to you. That's a really interesting question.
Interesting. So would you imagine that there's a difference between males and females for that matter?
So I'm not sure about the regeneration capacities, but I do know that the nervous system between the males and hermaphrodites in C. elegans are slightly different. They are male specific or hermaphrodite specific neurons. So so maybe they are regenerate similar on the shared neurons. Maybe those gender specific neurons may regenerate differently.
Thank you. Is there also an age dependency in terms of the levels of PARP expression or PARP expression?
That's a good question. So we know in the wild type animals, the axon regeneration declines as worm ages. So there's significantly reduced number of axon regeneration. For example, 5 days in 5 days adults. I'm not sure whether the PARP levels changes with injuries, but we do see that we do test in the PARP loss of function mutants in the aged animals, and we can no longer see the similar robust increase in terms of axon regeneration. So perhaps, the axon regeneration in aged animals is mediated by different mechanisms, or maybe the level of PARPs it also declines or increases in the aged animals.
Very interesting. Thank you. The next question is, from an evolutionary standpoint, wouldn't we expect axon regeneration be optimized in the wild type?
That's a good question. I always love the evolution questions. Again, my answer to this is that I actually think axon regeneration is actually inhibited in the wild type animals to prevent, for example, extensive regrowth or the wrong wiring of the regenerated axons to the existing nervous system.
Interesting. Thank you.
Yeah.
Can I just ask, so PARP expression in the human nervous system, how extensive is it? Is it the protein is conserved, right?
Yeah. So in worms, there's only 2 PARP genes, PARP 1 and 2. And in worms, in humans, the number of PARPs classes are much larger. I think there's at least multiple PARPs. So currently the PARP inhibitors is not able to efficiently target all of the PARPs protein in humans. Sorry, I missed the second half of your question.
Sorry, no, I was just asking as in how prevalent are these proteins in humans, and is that a similar pathway that is followed for axon regeneration or the lack thereof?
So definitely in the humans, the PARP mechanism is more complex, partially because there's more PARP type of class of PARPs present in humans. And also PARPs so the PARP is also implicated in different type of cancer. For example, the PARP inhibitors are very effective to treat certain type of breast cancer. So we're really excited to further explore this mechanism. Perhaps we can reuse what we already have, the FDA approved PARP inhibitors to treat neuronal injuries.
That would be very interesting.
Yeah.
The next question is, in aged worms, are they slower to regenerate axons than the younger ones?
So in terms of their regeneration capacities in the aged worm, those cell repair capacity definitely declines. I'm not sure, I don't remember in the top of my head whether the regrowth rate is also reduced in the aged animals, but I think the answer is probably yes. Both the regeneration capacities as well as the regeneration rate is reduced in the aged worms.
Interesting. The next question is, do worms regenerate under stress, for example, starvation or heat?
Yes, the answer is they can. I believe there's a study axon regeneration rate in the dower worms, which is the state of worms that worm will enter after prolonged time of starvations. I forgot the result of that, but people definitely study under different stress conditions. For example, heat shock or lack of food, whether the regeneration process is altered.
Thank you. We've got an amazing audience. We've got some really nice questions coming in.
Yeah.
So did you compare the regeneration capacity between normal and Alzheimer's model?
Okay, that's a really great question. I actually do not know the answer to. I can tell you that in the worms, we can express humanized Alzheimer-related proteins overexpressed in the worms, and then we can observe axon degenerates. But I'm not sure if at least I don't know. Sorry, on the top of my head, whether people injure those humanized Alzheimer's worm to study axon regeneration. Probably not, because if you overexpress those degenerated proteins, the axons already degenerate, so there's probably not that much axon to cut in those models. But I will definitely look into it and get back to you.
Interesting. So are there any PARP mutant mice available, and do they have a lot of neural defects?
Yeah, so as I mentioned earlier, there's a lot of classes of PARP. So for example, it will be a heroic cross to generate mice with mutants in different PARP mutants. We do study for example in the neuronal culture study I mentioned, we can use the sharpening RNA to knock down certain PARPs. And then we show that so far by knocking down one of the PARP1, we can see in vitro robust enhanced of axon regeneration.
It's really fascinating.
Yeah.
All right, I think we've got all the questions covered here. And this is an ode to you that we've got so many questions because the talk was amazing. So very nice food for thought here.
Okay, thanks, Wenjia and Carlos, for both of your exciting talks, and also thank everyone for joining in and all of your questions and discussions. And finally, we very much appreciate everyone who joined in for today's seminar to help us out by filling out our event survey. We will post that in the chat box.